electrocardiography. So it is called common abbreviation either EKG or ECG. EKG, that is the German version, and it is written as cardiogram, KARDIOGRAM, and most of the people here ECG is used. Okay, so basically it is the procedure of recording the electrical activity of the heart, and the electrode combination records the difference of potential bio biopotential difference at two sites on the body, and the potential difference are produced due to the electrical activity of the heart. And this electrocardiograph, which is the actually machine which is used to record that particular uh, electrical activity, and the record the graph which is coming after the measurement of this electrical activity is called electrocardiogram. Okay, so the characteristic shape and the timing of the ECG wave, these are due to the spread of wave of depolarization and repolarization associated with each heartbeat. Okay, so this is the heart and cardiovascular system. So it is referred as a two-stage pump which is physically arranged in parallel but with the circulating blood passing through the pumps in a series sequence. Okay, the right half of the heart, known as the right heart, is the pump that supplies blood to the rest of the system, and this circulatory path for blood flow through the lungs is called the pulmonary circulation, and the circulatory system that supplies oxygen and nutrients to the cells of the body is called the systemic circulation. Okay, so basically this is the cardiovascular system, which is actually known as the circulating system. So basically it transports the nutrients, gases, and wastes to and from cells of the body. So this cardiovascular system actually composed of
Excuse me, sir.
heart. Yes, hello.
A lot of students are trying to connect, trying to join the class, but they are unable to join. Some passcode is wrong, I think. They are facing it.
Sir, the meeting can't be joined if you are logged into Zoom. So you have to log out and then enter the Zoom.
Okay, I'm telling them.
Okay, so this cardiovascular system actually it is composed of heart, blood vessels, cells, and plasma, which makes up the blood. So this is the cardiovascular system representation, along with the this is the actually equivalent engineering type, which is called the piping diagram of the heart. Okay, what is given in here, the same thing is implemented. So this one and this one is, so this is the biological format and this is the engineering view. Okay, the piping system of the heart. So the actually, so it is a the pumping action here itself, it is performed by contraction of the heart muscles surrounding each chamber of the heart, and these muscles receive their own blood supply from the coronary arteries which surround the heart like a uh, crown. And this coronary arterial system, it's a special branch of systemic circulation. Okay, so blood actually enters the heart on the right side through two main uh, veins. One is the superior vena cava, which leads uh, from the body's upper extremities, and the inferior uh, vena cava, which leads from the body's organs and extremities below the heart. So the incoming blood actually it fills the storage chamber, which is called the right arteriole. Okay, right arteriole. So in addition to these two veins mentioned earlierly, that the coronary sinus also empties into the right arteriole. Okay, and the coronary sinus contains the blood that has been circulating through the heart via the coronary loop. So when the right arterium is full, it contracts and forces blood through the tricuspid valve into the right ventricle. Okay, then it contracts to pump the blood into the pulmonary circulation system. And when this when ventricular pressure exceeds the arterial pressure, this tricuspid valve closes and the pressure in the uh, ventricle forces the uh, semilunar pulmonary valve to open, and it causes the blood to flow into the pulmonary artery, which divides into the two lungs. In the uh, alveoli sides of the lungs, so there isn't some exchange. The red blood cells are recharged with oxygen and give up their carbon dioxide. Okay, the pulmonary artery, it actually bifurcates the many times into the smaller and smaller arteries, which becomes arterioles with extremely small cross section. Now these arterioles supply the blood to the alveolar capillaries, in which this exchange of oxygen and carbon dioxide takes place. On the other side of the lung, it is here the in which the capillaries feed into tiny veins or venules. It's called the venules. Okay, and it combines to form larger veins, which in turn combine until ultimately all the oxygenated blood is returned to the heart via the pulmonary vein. Now this blood enters into the left atrium from the pulmonary vein, and from there it is pumped through the mitral valve, or is called the bicuspid valve also, into the left ventricle by contraction of the arterial muscles. So when the left ventricular muscles contract, the pressure produced by the contraction mechanically closes the mitral valve, and the buildup of pressure in the ventricle forces the aortic valve to open, which causes the blood to rush from the ventricle to aorta. Okay, and so this action actually takes place synchronously with the right ventricle as it pumps blood into the pulmonary artery. So there are two terms you must familiar with. The heart's pumping cycle, it is divided into two major parts. One is the systole, another is the diastole. The systole is defined as the period of contraction of the heart muscles, especially the ventricular muscle, at which time the blood is pumped into the pulmonary artery and the aorta. And on the other hand, diastole, that is the period of dilation of the heart cavities as they fill with the blood. So once the blood has been pumped into the arterial system, the heart relaxes, the pressure in the chambers decreases, and the outlet uh, valves are closes, and in a short time the inlet valves open again to restart the diastole and initiate a new cycle in the heart. Now this mechanism this is happened in loop. Okay, so actually the heart beats at an average rate of about 75 beats per minute in a normal adult, and this figure can vary considerably. And the heart rate which increases when a person stands up, and decreases when he sits down. The range being it is from about 60 to 85. Okay, on the average it is higher in women, and generally decreases with age. And in case of infant, the heart rate may be as high as 140 beats per minute under normal conditions. Okay, so the heart rate that is also increases with the heart exposure and other physiological and physio uh, and psychological factors also, which is combined with this one to uh, increase of the heart rate. So heart pumps about 5 liters of blood per minute, and since the volume of blood in the average adult is about 5 to 6 liter, this corresponds to a complete turnover every minute during rest. And with heavy exercise, the circulation rate is increased considerably. At any given time, about 75 to 80% of the blood volume uh, is that is in the veins, about 20% in the arteries, and the remaining portion in the capillaries. Okay, so systolic blood pressure in the normal adult is in the range of 95 to 140 mmHg, with 120 mmHg being average. So uh, normal diastolic blood pressure, which is ranges from 60 to 90, and 80 is the average in case. Okay, and this pressure basically it is usually measured in the uh, brachial artery in the arm, as all of you have seen already. The pressure can be measured, how the pressure can be measured. Okay, so this is the internal structure of heart and the conduction system, which I have told you earlier. See the same thing. So here, actually, what happens in case of conducting system of the heart, which is the first is the SA node, which depolarizes, and electrical activity goes rapidly to AV node via internodal pathways from the SA node. Then from AV node, depolarization spreads more slowly across arteria. So conduction shows through the AV node, and then again depolarization moves rapidly through ventricular conducting system to the apex of the heart, and the last one is the depolarization of wave spreads upward from the apex, and it is continued. So this is the heart conducting system. So this man Einthoven, so actually there is a Einthoven triangle which is used to measure the um, ECG, or to use with the leads of the electrodes with the help of the formula which was invented by uh, William Einthoven. Okay. I will discuss it later, the intervening triangle. So what does the ECG look like? It look like this one. It's a graphical format over a paper. It is given in this way, and this wave has certain significance. What is the P wave? What is the QRS complex wave? What is the T wave? So it is the continuous form, and with the help of the measurement of each box of this paper or this strip, you can easily identify the status of the ECG recording. So normally ECG in ECG paper, so there are five boxes, one big box, which is equivalent to five small boxes over there, one, two, three, four, five. Okay, now this one is the time axis, this is the voltage. Now each one, this one, the small box, which is equivalent to zero point 0.1 millivolt. Okay, and this one is zero point zero 0.04, sorry, one box, the time axis, which is the 0.04 second. So the total time is 0.20 second, and the total millivolt is zero point 0.5. Is there any confusion regarding this? Okay, so next is the electrocardiogram waves. So this figure shows the ECG or electrocardiogram waveform as it appears when recorded from the surface of the body. Now, alphabetic designations have been given to each of the prominent, is given to each of the prominent features. These can be identified with events related to the action potential, which which is propagated. So it is that type of action potential propagation pattern. Okay, now to facilitate the analysis, the horizontal segment of this waveform preceding the P wave and P, which is designated as the baseline or the iso potential line. The P wave actually represents the depolarization of the atrial musculature. Okay, the QRS complex, this one, QRS complex is the combined result of the repolarization of the atria and the depolarization of the ventricles. Okay, now which occurs almost simultaneously. The uh, repolarization of the atria and the depolarization of the ventricles. Next is the T wave. Next is the T wave. That is the wave of ventricular repolarization, whereas the U wave, if present, it is not normally being there every time. If it is present, is generally believed to be the result of after potentials in the ventricular muscle. Okay. Now the PQ interval represents the time during which the excitation wave of these features vary with the so it is the excitation wave which is delayed in the fibers near the AV node. Okay, so that is the electrocardiogram wave. Now coming to the ECG amplifiers. So basically, normal electronic amplifiers are normally reference to ground through their power supplies. Now this creates an interference problem when such amplifiers are used to measure small bioelectric potentials. Okay. The technique which is usually employed not only in ECG but also in the measurement of other bioelectric signals in the use of differential uh, amplifier. Okay, so two amplifiers with separate inputs but with a common output is common output which delivers the sum of two amplifiers output voltage is a differential amplifier. All of you know that. So both the amplifiers have the same voltage gain, but one amplifier is inverting and while other is non-inverting in nature. Okay. So if the two amplifier inputs are connected to the same input source, then what happens? The resulting uh, common mode gain, that should be zero, because the signals from the inverting and non-inverting amplifiers cancels each other at the common output. Okay, so the gain of the two amplifiers is not exactly equal. This cancellation uh, is not complete. Okay, so a small residual common mode output remains always. So when one of the amplifier input is grounded and a voltage is applied only to the other amplifier input, the input voltage appears at the output um, by the uh, gain of the amplifier. Okay, and here it is use this second circuit. Okay, so the ratio of the differential gain to the common mode gain is called the common mode rejection ratio of the differential amplifier, which is in modern amplifiers that can be as high as uh, 110 lakhs is to 1. Okay, so measurements of these bioelectric signals that occur as a potential difference between two electrodes is an input to a differential amplifier. Now, the bioelectric signals are between the inverting and the non-inverting inputs of the amplifier, and for this, for the um, interference of signals, both inputs appear as they were connected together to a common input source. Okay, and a much smaller common mode gain amplifiers the common mode interference signal. Now, in this case, the electrode impedances, which is RE plus and RE minus, each form a voltage divider with the input impedance of the differential amplifier, as you can see in this figure. So, if the electrode impedance are not identical, what happened? The interference signals at the inverting and non-inverting inputs of the differential amplifier may be different, and the desired degree of cancellation does not take place. So why it is happened? Because the electrode impedances can never be made equal. Okay, the high common mode rejection ratio of differential amplifier can only be realized if the amplifier has an input impedance much higher as compared to the impedance of the electrodes to which it is connected. Okay. So next is the electrodes. Those are used in ECG measurement. So a number of electrodes, usually fiber affixed to the body of the patient for recording an ECG. Now these electrodes are connected to the ECG machine by same number of electrical wires. Now these wires which come from electrodes to the ECG machine are called leads. We will discuss it later after that. So the electrode applied to the right leg of the patient, as you can see in this picture, for example, it is called the RL lead. RL RL lead. Okay. For the recording of the electrocardiogram of two electrodes of one electrode and an interconnected group of electrodes are selected and connected to the input of the recording amplifier. Okay. Uh, so the pumping action of the heart, which generates the voltage, is actually a vector quantity where magnitude as well as orientation changes with the time, because the ECG signal is measured from the electrodes applied to the surface of our body. The waveform of the signal is very dependent on the placement of the electrode where it is connected. Now, some of the segment of this of the trace may uh, however almost disappear for certain electrode position, whereas other may show up clearly on the recording. For this reason, in a normal electrocardiographic examination, the electrocardiogram is recorded from a number of different leads. Usually 12 leads are used to ensure that no important detail of the waveform is missed. Okay. And this placement of that electrodes and the names and configurations of the uh, leads have become standardized. The color codes are also used to identify each electrode, as it is given here. You can see it. This was for RA, which is white in color. This is brown in color, black, red. This is green. Okay. So okay. So these are the abbreviations of and color codes of ECG electrodes. Now remember, the representation of the electrical activity of the heart is critically dependent on the position of the electrodes, as I have told you earlier. The same electrical activity will look different on different electrode combinations. And the combinations of electrodes for recording is actually called the lead. So 12 lead, the standard 12 lead ECG are used, and there are three types of leads. First one is the standard lib leads, which was invented by William Einthoven. Then is the augmented link leads, which is the Goldberger, and precordial leads, which are invented by the Frank Wilson. Okay, so these are commonly used leads for ECG measurement. So four electrodes are generally used to record the ECG. The electrodes on the right leg is only for ground reference, because the input of the ECG recorder has only two terminals. A selection of the, that particular selection can be made among the available active electrodes. So the 12 standard leads used most frequently, it is shown over here, and three bipolar limb lead selections first introduced by the Einthoven, which is shown over here. So here lead 1